به نام خدا. Biochemistry pharmacia, pyrimidine biosynthesis. First of all, I would like to ask you, what is the pyrimidine? It's a 6-member ring with 2 nitrogen in the meta position. She said E, nitrogen, meta position, okay? This is the base of the nucleotide base from the DNA. Which base is pyrimidine? Uracil, cytosine, thymidine. Now, what is the purine? Pyrimidine with imidazole ring, okay? I'm sure that you know the de novo pathway of the purine biosynthesis.
Yeah.
It says that nitrogen number 1 came from aspartate, carbon number 2 came from N10-formyl tetrahydrofolate. It means in the pharmacology, if we block the folate synthesis, the purine cannot synthesize because this 1 carbon came from tetrahydrofolate. Nitrogen number 3 came from glutamine, another amino acid. Carbon number 4 and carbon number 5 and nitrogen number 7 came from glycine, the simplest amino acid. Carbon number 8 came from N10-formyl tetrahydrofolate, again from tetrahydrofolate pathway. And the nitrogen number 9 came from glutamine. The purine ring structure can produce inosine monophosphate. The inosine produce adenosine triphosphate and guanosine triphosphate to RNA, and then if there will be deoxyadenosine triphosphate and deoxyguanosine triphosphate, they go to the deoxynucleotide triphosphate.
Purine inhibit here.
Yeah.
To inhibit
and azathioprine is the analog of that. Cytarabine is the analog of that. Fludarabine is the analog of adenosine. Yeah, these are the different drugs we have it on this pathway. The purine, now you understand it. The pyrimidine biosynthesis, the bicarbonate, it means HCO3 minus plus ammonium ion. It's used to ATP to consume the energy and produce carbamoyl phosphate. Could you please draw for me carbamoyl phosphate? Give me please one paper. Actually, that's the topic. Yeah. See? What is the carbonic acid? H2CO3. How can you draw the carbonic acid? You should put the carbon here, 3 oxygen around, 2 hydrogen. If they have a hydrogen, single bond. If doesn't have hydrogen, double bond. Carbamoyl phosphate, instead of 1 OH, you should put ammonium NH3 plus, and instead of 1 other OH, you should put OPO3 minus. Now, the carbamoyl is this one, with phosphate is OPO32 minus. This is a carbamoyl phosphate. Now, the pyrimidine ring, 6-member ring with 2 nitrogen, came from carbamoyl phosphate and aspartate. Carbamoyl phosphate and aspartate. What is the aspartate? Aspartate is an amino acid. If you draw it carboxyl, carbon, amine is glycine. Yeah? With 1 carbon is alanine. With 1 carboxylic acid is aspartic acid. How many carbon it has? 4 with 1 nitrogen. See? 1, 2, 3 with 1 nitrogen came from aspartate. 1 carbon, 1 nitrogen came from carbamoyl phosphate. And do you know where do we have carbamoyl phosphate? On the ural cycle. It means from the ural cycle, bicarbonate plus ammonium, and from the amino acid, and that's a feedback of the feeding pathway of the citric acid cycle, aspartate is come and pyrimidine ring produced. PRP, phosphoribosyl pyrophosphate, added to this group, and now we have a uracil triphosphate. First, we have a uracil triphosphate. From uracil triphosphate, we can produce cytosine triphosphate, and it go to RNA. From uracil triphosphate, by the help of the enzyme thymidylate synthase, we can produce thymidine monophosphate, and from the CTP also it will be deoxidized and the desoxycytosine triphosphate, and these two thymidine and cytosine go to DNA.
6-fluorouracil inhibit here.
5-fluorouracil.
5-fluorouracil.
Carbamoyl phosphate synthesis. I think we have another uracil analog also. 5-fluorouracil and. Please search about that. We have one more uracil analog. Azathioprine. No, azathioprine is adenosine. Carbamoyl phosphate carbamoyl phosphate synthase 2 on the cytosol. Okay, what's that? Bicarbonate HCO3 minus use ATP and phosphate comes in one of the O minus charge instead of OH minus. Phosphate minus came. Carboxyphosphate get the ammonia, and now we have a carbamic acid. Yeah. First the amine comes instead of phosphate. Phosphate is go away. Then to carbamic acid the ATP is used as energy, and the phosphate remain here, and now we have a carbamoyl phosphate. I drew it correctly, okay? COONH2 phosphate. Channeling glutamine. Glutamine has ammonia. Glutamine has always for the ammonia transfer we need the glutamine to glutamate, and ammonia is coming to carbamic acid, and then finally we can produce carbamoyl phosphate on this enzyme. What's that? Want to explain the aspartate. By the help of the aspartate transcarbamylase produce N-carbamoyl aspartate. See, where is the aspartate here? Carboxyl carbon amine is glycine, yeah? With carbon is alanine, with carboxyl is aspartic acid. This part is carboxylic acid. This, what's that? COONH2 is carbamic acid. Carbamic acid on the aspartic acid is carbamoyl aspartate.
Aspartate, carbamoyl.
Until here.
Carbamoyl aspartate.
You see, carboxyl carbon amine is glycine, alanine with carboxyl again is a aspartate. Now the enzyme dehydroorotase come, water is go away, and L-dehydroorotate produced. You see? Do you see NH2 and O? These two hydrogen of nitrogen with oxygen produce water. The nitrogen directly bond to the carbonyl group. Now this is the dehydroorotate. Dehydroorotate dehydrogenase. It will be dehydrogenized. And what's happened between these two carbon? Double bond form. Now this is the orotate. The orotate by phosphoribosyl transferase, phosphoribosyl pyrophosphate comes. The PPP is go away, and phospho with ribose comes here. Now this is the orotidylate. These are the multi-enzyme complex. The cytosine triphosphate can block this enzyme transcarbamylase on the aspartate and say that's enough, I'm here, please don't produce me again. CTP can, that's as a product, can inhibit. No, the orotidylate, this one, the orotidylate, by the enzyme orotidylate the carboxylase, the carboxyl, and now this is the uridylate, UMP. Oh, sorry. Without carboxyl, the carboxylase. The CO2 is produced. I think that's not important for memorizing. That's important to understand. Yeah. No, the uridylate, UMP, by the enzyme kinase, use 2 ATP and produce UTP. You see the 1 phosphate, now this is a triphosphate. 2 more phosphate is coming here. It's a uridine 5' triphosphate. Citidylate synthase. Glutamine produce glutamate. What is the difference between glutamine and glutamine? Glutamine has 1 ammonia. Glutamate doesn't have ammonia. Do you know what is the glutaric acid? You see 2 carboxyl. 2 carboxyl together is 2 کوه oxale, is oxalic acid. If I put 1 carbon here, it's malonic acid. Why? Because this carbon said this is malonic acid. If 2 carbon comes here, it's a succinic acid دوتا okay. If the double bond come here, that's a fumaric acid in the trans form, and the in cis position is the maleic acid چون مالش بینشونه. Now if I have a 2 carboxyl and 3 carbon, the name will be valeric acid, but with 4 carbon, the 4 carbon, if you eat 4 glue, your glue will be tariq. Tariq means dark, okay? Tariq means dark. If you eat 4 peach, 4 carbon, you see the glue in Persian is truth. Truth, sorry, I should translate it for you. But the 4, you cannot eat 4 carbon and it dark your truth, and the name is glue tariq. Lisa learned the sine and said, "Ma, succinic acid." This is the glutaric acid, yeah? Alpha ketoglutarate, this is a carbon. After the carbon is alpha, and this is also alpha carbon. Alpha keto has an alpha ketoglutarate acid. But now, if I, instead of alpha keto, I have an amine, this is not a glutaric acid, it's a glutamic acid. Yeah? If I think I add one more amine somewhere else, now this is a glutamine. Glutamine. Yeah. Now, it said glutamine comes and produce glutamic acid. ATP comes and just as an energy usage. And finally, the production, see what's the difference? It has a double O, it has an amine. Double O amine. Because the glutamine remain the amine here and get the carbonyl and produce glutamic. Now, from that's a cytidine triphosphate, and it can come back on the first reaction and set aspartate. That's enough. From aspartate, we produce what? You see, again, come back on this page. It's said on the pyrimidine biosynthesis, we need the carbamoyl phosphate and aspartate, and the production is carbamoyl aspartate, and finally it's produced, and UTP from orodate, what was the name? Orodate. From orodate, uridate is produced, and orotidylate, uritidylate. The UMP use the by the help of the enzyme phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate, but orotate phosphoribosyl transferase is coming here. UMP, UTP. Formation of UTP from UMP. Now the UTP is produced. The CTP synthetase, cytidine triphosphate synthetase glutamine. Come in, the glutamate go, double O produce double amine. From UTP, CTP is produced, yeah? Do you get this reaction? That's very important.
double O شده amine. اینجا گفتیم گلوتامین میشه گلوتامات.
I think you should remember also the shape of the nucleotide here. Yeah. Regulation by feedback inhibition. You see the CTP block here and said that's enough, please don't produce me. I'm here. And what was the name? Regulation by feedback inhibition on the pyrimidine synthesis. Please explain it. The CTP is produced and inhibit aspartate transcarbamoylase and inhibit the production of carbamoyl aspartate. Why it said N-carbamoyl phosphatate? Because on the nitrogen we have a carbamoyl. Nitrogen on the nitrogen, carbamoyl on the aspartate. Aspartate transcarbamoylase has an allosteric regulation. In this case, could be inhibited by CTP. Aspartate transcarbamoylase, ATCase. What's that? That's the Michael-Menten for this enzyme, yeah? If the substrate concentration is a lot, we can reach the Vmax. Normal activity, there is no any CTP. If I have a CTP plus ATP, I have a lot of ATP and I have a CTP also. The enzyme says, yes, I produce you, but I have a lot of energy. What can I do with this lot of energy? Nothing. In this case, I'm sorry for you, CTP. I should produce you more and more and more because I have ATP. But okay, if I don't have ATP and CTP is there, okay, this is the time of my rest. The velocity is decreased, the velocity of activity. Again, if you have a CTP and ATP, what's happened? There is a little change from the Vmax, a little bit decrease. Why? Because I have a lot of ATP. I should use ATP. You see, here again, in this pathway, where is ATP consumed? Here and here. Yeah. I have a lot of ATP. Okay, I would like to produce CTP, no problem. Because in this case, I can balance my energy. That's good. What's this? CTP synthase activity. The CTP synthase activity, if I have a lot of UTP, I have a Vmax. If I have UTP and CTP, it means the CTP is already produced a little. In this case, the rate of this enzyme, the activity decrease because, yes, I'm produced. That's not necessary to produce me anymore. Now, that one, glutamine plus bicarbonate plus ATP, PRPP, phosphoribosyl pyrophosphate. Who inhibit the PRPP? You remember we have such a drug that inhibit PRPP. They are very low. Is there any more? It should be a little bit more. The main card is not here. We have one oncology card that summarizes all of the drug there. See, not this one. Cholinergic agent. Hypertension, not. Antiplatelet. What's that? Anticoagulant, no. Let's see, all of them here. Asthma. Antimycobacterial. Antifungal. You see, all of the bacteria summarized here also. Antiviral now. Antiretroviral. This is all of the monoclonal antibody. All of the eicosanoid. Heart failure, diuretic. Oh, I don't have it. Why? After the PDF, I tell you. Sorry, I don't remember it now. Carbomoyl phosphate produce. You see, if I have a lot of PRPP, if I have a lot of phosphoribosyl pyrophosphate, phosphoribosyl, it stimulates this job and say, please please, active, a lot, consume us because we are a lot. If I have a lot of ATP, say, please, use the ATP. And the CTP inhibit here and here. There's a regulation of pyrimidine nucleotide synthesis. The ATP and PRP is stimulated, the CTP inhibited. It means this enzyme, carbonyl transferase on the aspartate, have allosteric regulation. UMP also inhibit orotidite monophosphate. I said, orotidite, please don't produce uridyl monophosphate. Now, monophosphate to triphosphates. AMP, it means adenosine monophosphate plus adenosine triphosphate by the enzyme adenylate kinase produce 2 adenosine diphosphate. Mono and tri produce dot D. Nucleoside monophosphate are converted to nucleoside triphosphate, yeah? Now, in this case, see, in the glycolysis, citric acid cycle, oxidative phosphorylation, the ADP can produce ATP, yeah? But never and never and never ADP plus ATP cannot produce ATP and ADP. Just AMP plus triphosphate can produce ADP. The ADP cannot combine with ATP and produce ATP and ADP. Mono and tri can produce D, but D and tri cannot produce another tri and another D. Nucleoside monophosphate plus adenosine triphosphate with non-specific of the sugar, that who is the sugar, ribose or deoxyribose, can produce nucleoside diphosphate and adenosine diphosphate from mono and tri 2 times D. It could be AMP, it could be NMP. The name of the enzyme instead of adenylate cyclase is nucleoside monophosphate kinase. What's that? Nucleoside diphosphate by the non-specific for sugar and the base plus another nucleoside triphosphate. Now it could be adenosine, it could be guanosine, anything. Can produce 2 times D. It could produce D and 3. It could produce from NDPA, NTPA, and NDPD. ADP, for example. You see, if you have an ADP and ATP with the same nucleoside, adenosine, you cannot convert it. But if you have a uracil adenine, you can produce uracil triphosphate, adenosine diphosphate with 2 different. But in this case, said, okay, from the first I was ATP, ADP. Now from the product I am also ATP, ADP. What is the benefit of this reaction? Now the name of this enzyme is nucleoside diphosphate kinase. On the diphosphate I want to add 1 phosphate. On the nucleoside monophosphate I can do the kinase job. Kinase means add phosphate. Add phosphate to NMP, add phosphate to NDP. Good? Salvage pathway. What is the salvage pathway? On the salvage pathway, the uridine plus ATP can produce UMP plus ADP. Just the uridine, just the nucleotide, the pure nucleotide, get 1 phosphate from ATP. The ATP will be UDP, ADP, and UMP is produced, uridyl mono. The cytidine, by cytidine kinase, CMP and ADP produced on the salvage pathway. First reaction. The second type is uracil phosphoribosyl transferase, uracil plus P or PP produce UMP plus PP. Uridine, cytidine, uracil. What is the difference between uridine and uracil? Uridine is just the nucleotide, but uracil, uridine is a nucleotide with ribose. Uracil is just the nucleotide, it's just a pyrimidine ring. See? If I draw it, 1 pyrimidine, double O amine, yeah? And here and here. See, now this is a uracil. If I add 1 sugar here, Yeah, yeah.
اسمش دبل او داره.
Yeah, sorry. If I add one sugar here, now this is a uridine. Do you get it? Yeah? Everybody get it? Uracil, uridine. Timidine. Cytidine. Plus PRPP produce now the ribose add and now the uridine monophosphate produce. We add the phosphor and ribose. You see, I have a uracil. I add the ribose with one phosphor here, and now this is a UMP, uridine monophosphate, and two phosphate. Yeah, and the two phosphate from. No, that's the question is. It has a 2-phosphate. The other 1-phosphate came from what? I think it should be derodin, yeah?
Pyrimidine pyrophosphate.
Yeah. That's a phosphor ribose. It has now we have already 3. Now the 1 and 2 is 3. Yes, that's okay. Ribonucleotide to deoxyribonucleotide. What's happened between them? Ribonucleotide are precursor of deoxyribonucleotide from RNA to DNA, yeah? Now, what's this? That's a pentose, ribose, phosphate, purine or pyrimidine. The carbon number 2, it has a OH or it has a H. It has a OH, that's a ribonucleotide. It has a H, that's a deoxyribonucleotide. Ribonucleotide reductase. We need ribonucleotide with this pathway. It is thyrodoxine, ferrodoxine, glutarodoxine, glutathione, NADPH is consumed to this pathway. And we have a drug that can inhibit the ribonucleotide reductase. Yeah? You have such a drug that can inhibit ribonucleotide reductase. نمیذاره از اینجا این دسته ها
اینجا که این وسط چیزی میکنه
چرا هیدروکسی یورئا این اینهیبیت ریبونوکلئوتید ریدوکتیز
جزء پیریمیدین چیزا جزء مکاپتوبوی هیدروکسی یورئا رو نداشت.
Yeah? If we have this pathway, the ribonucleotide deoxyribonucleotide is inhibited by hydroxyurea. That's a drug is used for which cancer? For sickle cell disease, for chronic myelogenous leukemia, as a cytoreductive therapy. The side effect is myelosuppression. That's a cell cycle specific chemotherapeutic. Cell cycle specific chemotherapy. No, that's it. Yeah. Ribonucleotide reductase has a regulatory one, regulatory another one subunit, regulatory 2 subunit, active site, ATP, DTP, DGTP, DTTP. You see a lot of substrate for primary regulation, substrate specificity. This is the allosteric effector regulatory site, active site substrate, with a lot of thiol as a thyrodoxine and gluterodoxine. Do you get it? A different site it has. Regulation of ribonucleotide reductase. How it will be regulated? ADP, GDP, UDP, CTP comes. The ATP consumed. Desoxyadenosine, desoxyguanosine, desoxyuridine, desoxycytosine produce. And from the ADP, the ATP produced, from the GDP, the GTP produced, from the UDP, TTP produced by thymidine synthase, and from the CTP, the CDP, the CTP produced. ATP is activate, and the adenosine triphosphate inactivated. The adenosine triphosphate is a final product. Said please, that's enough. Ribonucleotide reductase we produce already. Alteration of substrate specificity ADP تو دزوکسیادنوزین دیفوسفید این دیگه بلدیم دیگه. بلدی شما؟ ببین الان نگاه کن کی داره CTP رو استیمولیت میکنه؟ ATP. ولی کی داره اینهیبیت میکنه؟ TTP داره اینهیبیت میکنه اینو. و TTP داره استیمولیت میکنه اون یکیو. میگه منو ساخت تو رو بسازه. میبینی؟ میگه منو ساخت اینم نسازه. خیلی سخته. دیگه اینو یاد گرفتی دیگه. Thymidylate synthase اینجا مهمه. Where is the thymidylate synthase? Here. From DUMP to DTMP. From uracil monophosphate to thymidine monophosphate. Who inhibit thymidylate synthase? And 5-fluorouracil. You see 5-fluorouracil inhibit thymidine synthesis. 5-fluorouracil and also the prodrug of that capecitabine and fluorouridine. Floxuridine, capecitabine, 5-fluorouracil inhibit thymidine synthesis. Pyrimidine. We have a cytosine, thymine, and uracil. Yeah? The uracil has 2 double O, 2 carbonyl group. Thymine has a 1 methyl, and this methyl is came from tetrahydrofolate. And the cytosine has the amine different with uracil. It means from the glutamine, glutamate, it can be converted to each other. Thymidylate synthase. The folic acid decrease. In this case, uracil incorporation into DNA and DNA strand break. If we inhibit thymidylate synthase by the decreased number of folic acid, how? By the inhibition of the hydrofolate reductase or the dehydropetroate synthase. In this case, the folic acid decrease, uracil incorporation into DNA, DNA strand break. Yeah? And because we don't have a thymidine, the DNA strand is incorporated.
دیگه دیهیدروپتروئید سینتز مثل چیز دیگه
اینهیبیتوراش مثل سولفوناماید
و پیریمتامین و
پیریمتامین و تریمتوپریم inhibit the dehydrofolate reductase, not the hydropetroate. The DTTP formation, DUMP produce DTMP plus ADP, that's a thymidylate synthase. Then the thymidine kinase, the desoxythymidine to desoxythymidine monophosphate. This is a thymidylate kinase. This is a nucleoside diphosphate kinase, and the TDP to DTTP, thymidine triphosphate. We need tetrahydrofolate, NATP, methylene tetrahydrofolate, NADPH. Everything could be affected here. Biosynthesis of pyrimidine nucleotide. Please explain. We need carbamic and aspartate, and the pathway orotate, uridine, finally cytosine. Regulation of pyrimidine. Who can regulate it? Who stimulate it? ATP and?
ATP
carbamoyl. And who inhibit it? CTP. NTP formation from NMP. NMP produce ATP produce NDP. NDP plus ATP produce NTP plus ADP. Again. NTP formation from NMP. You see? NMP plus ATP produce NDP plus ADP, yeah? No, NDP plus another ATP produce NTP plus ADP. Okay? Salvage pathway for pyrimidine.
salvage pathway for pyrimidine که چیز شد دیگه همون
دو تا ریاکشن
داشت دیگه یکیش
uridine and cytidine plus ATP, UMP, CMP, or uracil plus phosphoribosyl pyrophosphate, not plus ATP, and produce UMP plus pyrophosphate.
uracil و uridine and cytidine.
اینجا ATP هست، اینجا PRP هست. But the first one is de novo pathway. This is a salvage pathway. Ribonucleotides conversion to deoxyribonucleotide by the help of which enzyme? Ribonucleotide reductase. And thymidylate synthase. Thymidylate synthase from DUMP. From DUMP produce DTMP. From uracil the thymidine is produced. Next video.